Well, if you've been here during this conference, I've introduced 2 times Dr. Graham Parker. Goes without, it goes without saying, he's really good. All right, I'll let him introduce himself in this panel. Graham, the platform is yours.
It was mine. There's going to be no jokes. There will be no humor. There's no humor. We're going to remove the needle, Bernie. Welcome. All of you people at the back, come over here. Come closer. We're going to tell you some stories. We're going to talk about adipose tissue. My name is, of course, Graham Parker. I am a real academic, but I'm also an editor. Editor of Stem Cells and Development, editor of Nucleic Acid Therapeutics, editor of Gene by Environment, and ethical advisor to Marianne Liebert Publishing. All of this is true. And everything you hear in this session is going to be true. We hear a lot at the World Stem Cell Summit about pluripotent stem cells. We hear a very small amount about adult stem cells, mature tissue-derived stem cells. But you're going to hear on this panel from 2 world experts on the biology of adipose tissue. And I'm going to read their introductions because otherwise I might not quite capture their full splendor. Furthest away from me, and still smiling, is Keith March, MD, PhD, FACC, who serves as director for the University of Florida Center for Regenerative Medicine. He's also the vice chief for cardiology research and professor in the University of Florida Division of Cardiovascular Medicine and the University of Florida Department of Medicine. Closer to me is Dr. Adam Katz. He is a professor of plastic and reconstructive surgery and serves as the director of the Plastic Surgery Research Laboratory right here at Wake Forest Institute of Regenerative Medicine. He also has appointments in the Department of Biomedical Engineering. Okay, let's try not to aim microphones at each other. Wow. I'm going to ask them some questions. Would any of you like to make any opening introductory remarks, or shall we just get to the questions as asked?
Let's just get to it.
You're happy to go for it? Okay. I'm going to ask Adam first what he considers to be the most significant advance in adipose tissue research that is now making it a more clinically relevant source.
Good afternoon, everyone, and thanks for having me be a part of this. Graham and Bernie, I really appreciate the honor to be a part of this. So the question was the biggest or most important advance in making adipose biology a clinically translational potential. My answer to that would be, imagine yourself back in 1990 or so when I was a resident. At that time, people had published quite a bit on extracting fat from animals, mostly mice, in the obesity literature. Rod Bell and others had described enzymatic dissociation. But in the world of plastic surgery at that time, believe it or not, we were moving and transplanting fat with fat grafting, and there was a significant amount of resorption of the graft. And so a major question at that time was, were there even viable cells at all in the liposuction? Because unlike the excision of fat from animals, the liposuction in humans is a fairly aggressive procedure, shall we say, where you're sucking fat through very small holes. And many people at the time thought that that process and the shear forces simply killed all the cells, and that's why the fat graft wouldn't survive. So I think at its very basic point there, the ability to show that there are viable cells that can be harvested, specifically from liposuction harvested adipose tissue from humans, was a major step, number one, and that could be done reproducibly. And the reason, of course, that it was important that it was in human and liposuction is because that's a very scalable method of obtaining large amounts of human tissue with no donor deformity. And I would say close after that, the second biggest part of that is not just showing that there are viable cells that you can harvest in liposuction tissue, but then the development of devices that can scale the isolation of those in a reproducible GMP type way.
Okay, thank you very much, Adam. Keith, you want to take a swing at this?
Well, Adam and I have been colleagues and friends for, the beginning of IFATS was when we connected, which is the International Federation of Adipose Therapeutics and Science, which he founded, and I got the privilege to join what was largely a group of people interested in doing and advancing at the very edge of plastic surgery, but I've always been there, the token cardiologist and vascular biologist. And so complementing the recognition within the world of plastics that Adam just described, I would say one of the most exciting revelations to the field that several groups found out in about the 2003 to 4 time frame, including our own, 3 groups I think published in that year, was that these cells are very useful in a world of angiogenesis. And this was related to the fact that they were and are found on the abluminal side of blood vessels. For a while we didn't know where the cells were in the fat, and we went looking for them, and we ascertained that. Now, we actually found the physical location of them later than when we realized what they could do, but realizing just by empirical experimentation and a crazy hypothesis, that both in vitro assays and ultimately in vivo assays, specifically, interestingly enough, all 3 of the groups involved, including our own, 2 were from Europe, 1 was us here. We looked at peripheral vascular disease models and found that these cells were amazingly good at revascularization. And it turns out it's good because they were at that, not because they were being coaxed to do something that they couldn't normally do, but rather what they were normally was basically part and parcel of blood vessels and vasculature within the fat, which is a very convenient place to get lots of blood vessels in the microvasculature. So I would say recognizing that brought the opportunity to mind to use these cells in areas that went further than plastic surgery, but across a whole platform of areas within the body. And then the second one I would add to that is realizing that the cells do a lot of what they do therapeutically by what they secrete. And then that basically created a groundwork for building the understanding across the field that the cells could do a lot of what they do, as I think has been heard earlier in this conference related to bone marrow, in their capacity as what Arnie Kaplan called the bone marrow compatriots of these cells, medicinal secretory cells. But these were things that we realized were paracrine in nature even well before that, and that paracrine activity, I think, is the other seminal finding that has underscored a lot of what happens in the field and what the cells can do, and now what their extracts or secreted factors can do.
Terrific, thank you very much. It would be negligent to have you wonderful clinical researchers on this stage not address what your current favorite projects are that you're undertaking that are relevant to the topic we're discussing today. So what's getting you out of bed in the morning at the moment, Adam?
Yeah, good. Okay, favorite current projects. There's probably 3 for me. One by far is my non-day job association and disclosure is being part of a company trying to commercialize adipose-derived cell therapies is the GID Bio Phase 3A pivotal trial using autologous SVF cells from adipose for knee osteoarthritis. So we are over halfway through enrollment of that pivotal trial, and all the data is blinded, so can't really comment on that. But the phase 2 data was very encouraging. Number two is in the lab. We have 2 projects in the lab I'm excited about. One is really a basic thing, going back to how many cells are in a gram of fat. And no pun intended, gram, but that's really an interesting question. And what we're finding with different techniques is that the SVF harvest that we do, that we thought, I think the field believes we're extracting most, if not all, of the stromal cells from adipose tissue, is completely wrong. We're leaving a lot of chips on the table. There's an enormous number of non-adipocyte cells left behind in the enzymatic residue, what we call scientifically the snot. And so I think there's some really interesting pathways forward there. And the third is a single-cell RNA sequencing project where we're trying to really understand better the different subpopulations within the stromal vascular fraction and how they interact and where they localize after implantation.
Okay. If it's not a rude question, Adam,
Yes, sir.
who's funding your trial?
The clinical trial is the company. GID Bio Incorporated is a private entity funding the phase 3 trial.
Oh, wow.
Yeah.
That is exciting. Okay, Keith?
That's an interesting question. That's a hard one in a way because there are multiple things we work on. But I think I can probably parallel Adam's discussion in some way. Not in this order necessarily, but one of them to be parallel purposely. We have biotech that we spun out of our group a number of years ago, Theratome Bio, and its fundamental purpose is to bring forward the platform of secretome-based material for a variety of indications, and it's in fundraising mode now with effectively a post-pandemic reinvigoration, restarting. It has had the benefit of Series A funding and also a number of federal grants, SBIR and DOD in nature. But bringing these now into clinical trials is where we are right now with prioritization of that going on. So that's exciting to see finally moving forward again. On the academic side, our major focus there for a number of years has been and continues to be looking at the possibility that the secretome not only is useful in the in vivo setting clinically, hopefully, as it is in many of the preclinical or non-clinical experiments we've done with a variety of disease models, but also ex vivo to help with organ or other tissue, solid or even liquid transplant preservation. The heart is our key focus since I'm a cardiologist. It turns out that the heart is able to travel under cold ischemic transport conditions, as we heard in this conference actually in a couple of different presentations over the last couple days. In a limited degree. It can only travel typically 4 hours before it's not very often accepted as an organ for recipients. And the reason for that is because of the incrementation of immediate or subsequent subacute organ failure post-transplant, because it's basically had a whole heart attack on the way. Right. What we found is that the secretome being used in admixture to the current clinically used transport media can actually extend the ischemic tolerance of that to perhaps as much as 8 hours when we look at human cardiomyocytes beating in culture. And now we're moving, as we've done that already in rodents and looked at the mechanisms as well, we're now moving that into pig hearts, and we hope to move it into human hearts. And so this is the topic of funding from the VA and a number of other sources over the years. So we're really looking at, can we use secretome ex vivo as well as in vivo, and can we use it to really expand the amount of organs available? Amazing statistic, 3,000 hearts are transplanted roughly per year, 3,000, 3,500 over the last several years. That is out of a pool of 10,000 hearts that become available through a donor tragedy. 7,000 or 70% of the hearts are thrown out in the garbage. And about half of that wastage is because of this problem of not having enough tolerance to ischemic transport. So our take on that is, let's put some freeze-dried secretome in the hands of the transplant surgeons and have them admix it. The third one is a very similar topic, and it has to do with strokes. And again, one of our visions is to put, not only for stroke, but other kinds of very specific traumatic brain injury and others, but basically in CNS disease, to try to place secretome, whether frozen or freeze-dried, into ambulances and emergency rooms. And we're working on some grant funded as well as translational things through the biotech on that as well.
Fantastic. I hope the audience is thinking of some questions to ask the panelists. When we do these presentations, I think there's always this temptation, and I think you both may be a little bit guilty of this. It sounds like everything is just going so great. Everything's getting solved and everything is moving forward in the right direction. I wonder if you both would care to comment on what you see as being what's the remaining hurdle to advancing the use of adipose tissue, both in reparative surgery, but also beyond that to other indications, as Keith has been discussing.
I'd say the hurdle oftentimes really is not in the concept or not in what we see biologically, but in finding the funding, whether by grant mechanisms or by Valley of Death traversal for a startup, whatever world you're in, or both, to actually go into humans and do a meaningful trial. I mean, this is probably not a rocket science answer, but it's true. If we can move with shots on goal into actual clinical trials and show what's happening there, that will help us to focus where our next step should be. But that's a major hurdle. I mean, I don't know if everyone would have experienced this, but being in an academic world over many years, as well as in startup or in sort of that kind of translational approach, much of the time is spent not doing data or running trials, but finding the resources with which to do it. It's not the thing we are really prepared for when we come out of all of our training and want to invent things, but that's what we do. So that's a big hurdle. Another hurdle, practically, you would hear from, I think, a regulatory perspective, is being able to handle the fact that whether you're working with an allogeneic approach or whether you're working with an autologous approach, in both worlds, consistency of preparation and how to demonstrate its potency becomes a very significant consideration, and figuring out enough of the biology to be able to do that is important. And the third thing I would say, besides that potency issue, is there a way to predict a little more accurately than not at all who might be a responder in any given scenario? We know in all of medicine that not everybody responds with a significant benefit to any given therapy. And as a cardiologist, I mean, I think back to the thrombolytic trials in the late 80s, early 90s, even into the 2000s, and so many versions of TIMI and TAMI and streptokinase, and then later on TPA and urokinase. These trials recruited 10,000 people or 20,000 people typically, and sometimes they would show benefits which were accepted by the field as important, but the benefits were in the single-digit percentages of hard endpoints, as they're called, or surrogate endpoints. And so you get around then to the idea of the number needed to treat, and NT. And there's like, you have to treat 10 people or 8 people or 200 people, not really, but usually a pretty sizable number to treat to get one person some significant clinical benefit. largely because that way of thinking isn't present and the funding and resources aren't present. We're not treating the cell therapy or cell-based therapy field in quite that same way. People expect a miracle, as it were, where you treat one and you get one great result every time, or even three and you get one, which is maybe more practical. But you know, the statistics to find those numbers needed to treat when you're in the domain of what we see routinely in cardiology are that you have to invest a lot more effort into larger trials and not expect miracles, but advances. That would be my threefold.
Thank you.
Yeah, of course, I would echo many of the things Keith said. And as you questioned, I mean, I certainly hope we don't make it seem like everything is easy and goes well. That's far from it. It's very difficult to take an idea in good science. Even really great science is difficult to translate. It's even harder to commercialize. And in my mind, there's a big difference. You know, translating something into humans in clinical trials does not mean it will be a commercial success. If it's not going to be a commercial success, I hate to say it, if there's not going to be money to be made by someone, then that someone will not invest their money and put it at risk. That's what makes treating orphan drug markets and small markets difficult. So there's no question raising money is incredibly difficult. We're experiencing that now. We're in a phase 3 trial, and it's still hard. And it's hard because of multiple factors. I would have said, 5 or 10 years ago, what the hardest obstacle was the regulatory path. But a large reason for that at the time was because the regulatory path wasn't clear. So it was hard to write a business plan with the next steps being very clear for an investor to say, oh, I'll get behind that. Now the regulatory path is very clear. It's just very long and very hard and very expensive, but it's clear. I would also say that, running clinical trials is incredibly difficult. Enrollment takes time. It's very difficult. And running a clinical trial for a pivotal FDA commercial objective is also very different from running an investigator-initiated clinical trial that's looking to expand the world of knowledge. Two different beasts. So for me, there are enough cell therapies and even adipose cell therapies that are in phase 3 or soon in phase 3 and going along the FDA path. So in the end, what the field needs, and I say this for all of cell therapy, the field needs one really great winner to move the field along. And by that, I mean something that is therapeutically proven to be efficacious at a cost-competitive price point to the standard of care with a reimbursable CPT code. And without any of that, some things are going to be translated, but they're not going to be commercial successes, which means they won't impact the masses that we're trying to help.
Okay, I would push back a little bit on that though, because if you think of an example from a different field, the development of Spinraza, an incredibly expensive medication for a relatively small patient number. The power of that, though, was that it showed then a model that could then be followed for future development, as well as educating the FDA and pushing things forward. So I'm not sure it always has to be the way you're describing.
No, I agree with you. I think that's one pathway, and with many of the incentives for small markets and rare diseases, I think that's fantastic. I mean, my world, Botox, like one of the most used poisons ever in the history of mankind for therapeutic purposes. That was a very specific orphan drug market for blepharospasm. Now it's used for everything. Every aging wrinkle on your face, it's approved. And they did it in that way. So that's definitely one path to do it. But I was thinking more in a broader scheme, like, I mentioned we're in osteoarthritic trials. Those are high hurdles to show that you're better than the standard of care. And if you're better, that you're cheap enough to be worth it.
Okay.
Oh, yeah. Yeah, that's a key thing he said at the end there, cheap enough. I think if we can get these agents down to the point where they're, single-digit thousands of treatments, that's a way dollars per treatment. That's a way different world of accessibility and affordability and like people accepting it. Acceptability maybe is a better one, but and I think we should be able to get there at scale and with various manufacturing improvements. I've heard of people talking about that and maybe experiencing it, but when something that we know almost certainly is going to have to be repeated multiple times, because these are not typically engraftment phenomena. Some are, but the ones we're talking about here are not typically engraftment based, but rather modulation based. This might be yearly or quarterly or whatever, some number of therapies. They need to be in the thousands range, not the tens or hundreds of thousands. So I think manufacturing at a level that allows that to occur is going to be a very, very important contribution.
So my last question before we can open up to the audience. I've had the privilege of working with a great many different stem cell types. And I think of adipose stem cells as still being pretty much in their infancy in terms of their characterization. Obviously, in terms of their clinical use, one cannot be manipulating the source tissue. But would you guys like to speculate on whether having a more mature and developed understanding of the adipose stem cell might serve us well? I'm thinking of the comparison to, you mentioned the stromal fraction that you were dealing with. I'm thinking of the hematopoietic stem cell that has been so intensively investigated and characterized. Whereas the adipose stem cells, I'm just not seeing many papers submitted to my journal that are actually looking to characterize the adipose stem cell. Is that a fair comment?
Yeah, I think it's fair. I think it's fair to a point, but yeah, those of us who are interested in translating it to help people, we're interested in doing the minimal essential research to get it into human trials. And then the challenge is to show that it's safe in human trials, right? That is a far cry from understanding the mechanism of action of how things might or might not be working. No question about it. But interestingly, when you get to the FDA, that's not where they push back, right? But as a scientist, by all means, the more we know about it, the better everything will be. But as a caveman surgeon, if it's safe in humans and effective, it's like aspirin. We didn't know how that worked for quite a long time. But in the end, I can't disagree. There needs to be more basic science and more sophisticated science that helps us understand characterization, mechanism of action, engraftment, all of that. Yeah. And we need funding for that, Graham.
I understand. This was cheeky of me because they didn't know I was going to ask them that. It was a cheeky question.
Well, I would agree. The aspirin comment was a good one, and the bark where it came from originally. But directed to cell therapy, I think most of you who know about the history of bone marrow and cord blood transplantation would recognize that it saved a lot of lives. It hasn't been able to save everybody either, but I think in 2013 was the year when we crossed 1 million such transplants, if I remember roughly. And at that point, starting from the 70s and 80s, when those approaches began and were progressively refined, until maybe the last decade, we had no idea what was the most uncontestable surface marker of the truly regraftable and highly proliferative hematopoietic stem cell in the SLAM receptor set or family. So it took a very long time until that was identified, which was a key part of how to give the cell its identity and even functionality. If people had held off to wait until they understood it, that would have been a delay of 30 or 40 years of quite a few 100,000 lives saved in the world of cell therapy. So I'm all for understanding and even more all for moving things forward to learn what actually works ultimately in the clinic. So he said he's a caveman surgeon, and I'm just a country cardiologist.
Okay. Well, we're forming a band up here. Are there any questions from the audience? Ian, could someone bring Dr. Waiter a microphone? Thanks, Joe. Could someone bring him a working microphone?
Okay, it's working now. I debated whether or not to ask the question because it's not really my place, but I figured seeing as you asked. But it is sort of pertaining back to the mechanism of action. You had discussed trying to figure out whether the aspiration itself led to shear stress and the death of the cells, but it's always been my understanding, and again, I'm not an expert like either of you two gentlemen. It was always been my understanding that SVF, like bone marrow, acts, and like PRP, acts mainly as an irritant. Is there a lot of evidence to suggest there's a difference in outcome between shear stress dead cells versus live cells? And if so, which of the cells in the SVF are you think engrafting? And I don't recall seeing much evidence for the engraftment, but could you comment on whether you think there's an aspect of the irritation, the cells degranulating, the cells apoptosing in the joint, initiating an immune response, leading to repair, versus the cells themselves acting directly? I hope that's it. Okay.
Yeah, I say 100%. I would, instead of use the word irritation, I would use inflammation. And I would say 100%. We have some murine model data where it's actually, you don't even need the cells from adipose. There's oil that gets broken down into free fatty acids, and those can generate a really prolific inflammatory response in the peritoneal cavity, for instance. But what does that mean under the skin for a wrinkle? Pro-inflammation could be a really great thing to help collagen remodeling and regeneration of the skin appearance. So I'm totally on board with you. I don't know if it's the dead cells. I don't know if it's live cells or which subset. I feel very strongly it's a lot of oil in the fat grafting and fat that we do for some of the therapies we're trying.
I don't know that I have anything to add. I think that was very good.
Beautifully answered, Adam. Well, I would invite the... Oh, there's a man with his... Do we have time for one more? Yes, thank you. A microphone is coming to you, sir.
Any comments to share on brown fat, beige fat, and the therapeutic use, or maybe just the need to study it more since you have access to a lot of fat? It seems to be a hot topic, especially for diabetes, weight loss, and also angiogenesis.
Well, it's certainly a very good type of fat to obtain, and people are finding in general that it's more difficult to, well, we know it's more difficult to find those. Not everyone has them autologously anymore as an adult. If we can brown them, essentially create a tanning bed for fat, that would be wonderful. Just made that up. But I think we need to brown the fat on the ex vivo side for it to be generally available on an autologous side. On the other hand, if through IPS mechanisms or others, we can create either allo or autologous conversions, probably allo would be more practical. The brown fat engraftment is going to be, I think, a different story because I don't believe, we haven't done this work in specific, and we worked on it a number of years ago, but not lately. I don't believe that necessarily its therapeutic effects are more mediated by secretome, but rather by the assistance of energy consumption being driven by the mitochondrial circumstance with the, with the different pores that it effectively has and the uncoupling. So consequently, I think it's going to be necessary to engraft brown fat chunks, if you will, or colonies, much like an islet is engrafted. So here again comes a subservience of the regular ASCs perhaps and their endothelial partners. I can see, little brown graft implants for sure. If that's a very viable approach, but it's a very distinct approach from everything else we've been talking about.
Yeah, I'll just quickly add, I don't work in that field often. I think it's an exciting area that's ripe for innovation and translation.
Terrific. All right, ladies and gentlemen, please thank Keith and Adam for a very exciting session. Enjoy the rest of your day.
Well, we are all adipologists now, adipology. We've all learned something. We will have t-shirts printed with that adipology. I never appreciated fat quite so much as that panel. Thank you, Dr. Parker, for chairing the session.